Hi, I'm Antonio, and I'll talk about our paper, Variational Combinatorial Sequential Monte Carlo Methods for Bayesian Phylogenetic Inference. This is joint work with Lee Zhang, Christian Nassif, Hattie Venner, David Bly, and Itzhak Pierre at Columbia University. We'll introduce a powerful framework that establishes variational sequential search to learn distributions over intricate combinatorial structures. We'll sketch an efficient proposal distribution, which provides an exact approximation to the intractable locally optimal proposal, and we'll use that proposal to define a second objective, which yields tighter lower bounds. At its core, phylogenetic inference aims to understand how life evolved over time. The goal is to use observed molecular sequences to reconstruct evolutionary history, which we can think of as a latent bifurcating tree. More specifically, a phylogeny is defined by two objects: a discrete topology, which is a connected acyclic graph, an instance of a bifurcating tree with a set of nodes and edges, where leaf nodes are the observed taxa, internal nodes are the ancestral taxa, and the root node is the common evolutionary ancestor. For each edge, we associate a continuous branch length exponentially distributed that represents time until a coalescent event or time for a mutation. We're particularly interested in learning distributions over composite structures that include both discrete and continuous spaces. Given a phylogeny, topology and set of branch lengths, and an alignment of observed molecular sequences, we need a model to denote the likelihood. While our setup can work with different generative models, it's useful to think of a specific model. We assume the likelihood of the data is independent across the M alignment sites, and we can use a continuous time Markov chain to model evolution, where the probability at site S of V mutating to V prime is given by multiplying the branch length and the transition matrix, and then taking the matrix exponential. We can write this out in full and note that the tree likelihood can be marginalized in linear time using Felsenstein's pruning algorithm, which is a special case of sum product or belief propagation for bifurcating trees. We simply pass messages from leaf to root, from left and right child nodes to the parent. One of the main challenges is that the number of rooted binary trees scales double factorially with the number of leaf nodes. Here's an illustration of the 2n minus 3 double factorial, or 15 distinct topologies for 4 leaf nodes. We can write the posterior over topologies and branch lengths to model evolutionary uncertainty. And we find that computing the likelihood involves an intractable summation over topologies and an intractable integral over branch lengths. It's useful for us to distinguish these two computational tasks required for phylogenetic inference. The first task, inference, involves computing the normalization constant by marginalizing 2n minus 3 double factorial topologies. A common approach used for approximating is to sample topologies and branch lengths via Monte Carlo methods, given that parameters are known. The second task, learning or optimization, refers to finding the set of parameters that maximize the data likelihood obtained by marginalizing the joint density. Sampling algorithms can also be used by assigning a prior to theta, the parameter set, and then performing a local search for the parameters via MCMC methods, given the data likelihood is available. Given that this is a composite space with multi-modes, we're often facing long runs and inefficient state space exploration using MCMC methods. Sequential search, on the other hand, factorizes the distribution into this sequence of probability spaces. K intermediate objects called partial states are maintained as forests over the set of taxa, and trees are sampled and built iteratively from leaf to root in a process involving 3 steps, which we sketch. First, K partial states are resampled to focus on areas of high probability. Then a new partial state is proposed by sampling 2 trees in each forest to coalesce uniformly and simulating branch lengths. Finally, an importance weight is assigned to a new partial state by using the pruning algorithm to calculate the likelihood. This process yields an unbiased estimate for the marginal likelihood that converges in L2 norm. The idea of our method is to use CSMC as an approximating distribution, which in turn can be used to form a variational objective. So we introduced the first method that uses sequential search to learn distributions over composite structures, combinatorial structures. A drawback of the CSMC method is that trees are sampled uniformly to coalesce when many of the resulting topologies may correspond to areas of low probability mass. We can think of a locally optimal proposal that uses information from one future iteration in order to guide the exploration of partial states. This locally optimal proposal requires us to marginalize over intermediate topologies and branch lengths. We developed nested CSMC or NCSMC and proved that it gives an unbiased, consistent, and exact approximation to this intractable locally optimal proposal. So for a given partial state, we enumerate all n minus r choose 2 look-ahead topologies. For each potential topology, we subsample m branch lengths to marginalize over that space. In the example here, we only show one subsample per intermediate topology. Finally, we compute a twisting potential to each partial state, which is then used to sample the chosen partial state from the candidate set. We choose nested CSMC to define a second approximate posterior and objective, which we call VNCSMC. We run our method on a benchmark data set of primate mitochondrial DNA over 12 taxa with over 13 billion distinct topologies and 898 sites, reporting the log marginal likelihood values, the running times, and the effective sample sizes. We show VNCSMC with K equal to 4 gives higher values than VCSMC with K as 128. We also show near optimal effective sample sizes for all choices of K, the number of samples in both methods. Here we briefly compare the maximum likelihood phylogeny of Mr. Bayes and VNCSMC and find similar topologies. The bottom clade partitions monkeys and the central and top clades partition hominids and prosimians respectively. For context, Mr. Bayes uses 20,000 iterations of MCMC in contrast to VNCSMC, which only uses 256 samples. Here we run our method on 17 species of Betacoronavirus data over 36,000 sites and show a phylogeny. Colors denote species of 4 viral lineages: Empicovirus orange, Nabicovirus blue, Sarbecovirus red, including SARS-CoV-1 and 2, and Rabicovirus in gray. To conclude, we've introduced the first discrete variational sequential search algorithm to learn distributions over combinatorial structures, establishing connections between VI and sequential search. Our method is applicable to other models outside of phylogenetic inference. For more details, please see the paper. Thank you. Implementation.